Fertilization depends upon the two gametes bumping into one another. In species with internal fertilization, which includes all mammals and birds, both sperm and egg must be transported into the oviduct, which serves as the site of fertilization. Sperm transport. Semen is ejaculated and deposited initially into one of two sites, the vagina, that is in humans, cattle, rabbits, or the uterus, example your horses, pigs, rodents. In species such as dogs, semen is probably deposited largely into the vagina but also forced into the uterus. Despite these differences in deposition site and significant differences in the number of sperm ejaculated, there is remarkably little variation among species in the total number of sperm that reach the oviducts. Typically, a few hundred to few thousand sperms reach the oviducts following a single mating, which usually represents far less than 1% of the sperm in the ejaculate. The vagina represents a hostile environment for sperm, and their continued survival depends on getting into more hospitable regions of the female tract. In their journey from vagina to oviduct, sperm must overcome a series of barriers, each of which eliminates a substantial proportion of the original population of sperm. The cervix connects the vagina to the uterus. The cervical canal follows in irregular tortuous route, and the epithelium contains many deep crypts. The cervical epithelium is richly endowed with mucus secreting cells, and as a consequence, the lumen is filled with mucus. Interestingly, the consistency and viscosity of cervical mucus is under endocrine control. When estrogen levels are high and progesterone levels low, as occurs prior to ovulation, cervical mucus becomes watery and its mucin strands assume a parallel orientation. This state apparently greatly facilitates passage of sperm through the cervical canal. Conversely, when progesterone concentrations are high, as in the luteal phase of the cycle, cervical mucus becomes exceptionally viscous and disorganized, which largely precludes entry of sperm into the uterus. The uterus doesn't present an active barrier, but sperm must somehow be transported directionally along its length. Studies in several species have shown that sperm are able to get from the distal uterus to the oviducts in times as short as few minutes, which is much too fast to be explained by sperm motility. Moreover, dead sperm and innominate sperm-sized particles are rather efficiently transported upward through the uterine lumen. The conclusion from these types of studies is that sperm transport in the uterus is largely a result of uterine contractions, and that sperm motility plays a minor, if any, role in the process. In most but not all species, the uterus is also a site hostile to sperm. In many animals, sperm within the uterus are rapidly phagocytosed. In other cases, sperm can remain viable in the uterus for several days, but their fertility rapidly declines. There are some dramatic exceptions to these general observations. The uterotubal junction is the region joining the tip of the uterine horn to the oviduct. The morphology of this region varies considerably among species, and this structure appears to be significant barrier to sperm, especially in animals like rodents and pigs, where huge number of sperm are deposited directly in the lumen of the uterus. In summary, the vast majority of ejaculated sperm are lost at various points between the cervix and oviduct. A few exhausted semifinalists make it to the site of fertilization. Of those, of course, there can be only one winner for each egg. Without meaning to, John Waynes provided a good synopsis of the life sperm. Mammalian eggs are ovulated from ovarian follicles as cumulus oocyte complexes, which consist of the oocyte embedded in the cluster of follicle cells. Here you can see this in this image, the oocyte is encased in its zona pellucida, which is somewhat obscured by a cloud of follicle cells. In order to reach the site of fertilization, the ovulated egg must be picked up and transported into oviduct through an opening called the ostium. In most mammals, the ovarian end of the oviduct flares into a funnel-shaped structure called the fimbria, which is positioned to partially over the ovary. The fimbria is densely covered with ciliated epithelial cells, which beat towards the ostium and propel the cumulus oocyte complex into the oviduct. In species such as the rodents and dogs, the ovary is enclosed completely or nearly completely in a thin membrane called the bursa, because the ostium of the oviduct is inside the bursa. The eggs are essentially trapped after ovulation with nowhere to go except into the oviduct. Once an oocyte enters to the oviduct, it is propelled by ciliary motion down into the ampulla, where fertilization makes. The oviduct provides the appropriate environment not only for fertilization but for early embryonic development, and it is important that the embryo remain there for a period of about 3 days. The fertilizable lifespan of gametes. In most species, both sperm and egg have a short fertilizable lifespan, and once they are delivered into the female tract, the clock starts ticking. What this means, of course, is that mating or insemination must coincide closely with ovulation. If sperm are deposited many days before the egg reaches the oviduct, there is little chance that they will survive to fertilize. Conversely, if sperm reach the oviduct several days after ovulation, they will certainly find an egg that has long since degenerated. In humans, the ovulated eggs has a very short fertilized lifespan. Fertilization presents some major challenges to both sperm and egg. The fertilizing sperm must somehow recognize, bind to, and ultimately traverse the zona pellucida surrounding the egg. It then must bind to the plasma membrane of the egg. The egg must not only respond to the fertilizing sperm in a number of ways, but actively prevent more than one sperm from fertilizing it. Fertilization by more than one sperm is bad. In their mature form, both sperm and egg possesses structures that allow them to fulfill these mission objectives. We'll see about the structure of the sperm. Mature sperm, known formally as spermatozoa, have a morphology that most people over the age of 10 would recognize immediately. The nucleus is contained within the head. which are most mammals has a flattened oval shape during the spermatogenesis. During the spermatogenesis, the haploid sperm and the sperm cells develops a tail or flagellum, and all of its mitochondria becomes aligned in a helix around the first part of the tail, forming the midpiece. The entire cell is of course enveloped by a plasma membrane. So the other structures in the mature sperm that plays a critical role in fertilization is the A chromosome. The A chromosome is in essence a lysosome that forms around the anterior portion of the nucleus. It is bounded by a membrane that is considered to have two faces. The inner acrosomal membrane faces to the nucleus, while the outer acrosomal membrane is in close contact with the plasma membrane. Structure of the egg. Most mammals ovulate an egg that has matured into a secondary oocyte. It is always the secondary oocyte that is fertilized. The secondary oocyte is produced along with the first polar body as a result of the first meiotic division. Both of these cells are encased in a thick glycoprotein cell called the zona pellucida. Genetically, the secondary oocyte that arrives in the oviduct is in metaphase of the second meiotic division. The metaphase plate is located inside the oocyte immediately below the first polar body. The final structural feature of the egg that serves a critical function during fertilization is a set of cortical granules. During oogenesis, the oocyte develops thousands of small membrane-bound granules that accumulate in the cortical cytoplasm just beneath the plasma membrane. What is fertilization? Fertilization is more a chain of events than a single, isolated phenomenon. Indeed, interruption of any step in the chain will almost certainly cause fertilization failure. The chain begins with a group of changes affecting the sperm, which prepares them for a task ahead. Successful fertilization requires not only that a sperm and egg fuse, but that not more than one sperm fuses with the egg. Fertilization by more than one sperm, polyspermy, almost inevitably leads to early embryonic death. At the end of the chain are links that have evolved to efficiently prevent polyspermy. In overview, fertilization can be described as a freshly ejaculated sperm are unable or poorly able to fertilize. Rather, they must first undergo a series of changes known collectively as capacitation. So, what is capacitation? Capacitation is associated with removal of adherent seminal plasma proteins, recognition of the plasma membrane lipids and proteins. It also seems to involve in influx of extracellular calcium, increase in cyclic AMP, and decrease in intracellular pH. The molecular details of the capacitation appears to vary somewhat among the species. Capacitation occurs while sperm reside in the female reproductive tract for a period of times, as they normally do during the gamete transport. The length of time required varies with the species, but usually requires several hours. The sperm of many mammals, including human, can also be capacitated by incubation in certain fertilization media. Sperm that have undergone capacitations are said to become hyperactivated and, among other things, display hyperactivated motility. Most importantly, however, capacitation appears to destabilize the sperm's membrane to prepare it for the acrosome reaction. So, how this occurs? Sperm zona pellucida binding. First thing. So, binding of sperm to the zona pellucida is a receptor ligand interaction with a high degree of specificity. The carbohydrate groups on the zona pellucida, glycoproteins, functions as sperm receptors. The sperm molecules that binds this receptor is not known with certainty, and indeed there may be several proteins that can serve this function. The acrosome reaction. Binding of sperm to the zona pellucida is the easy part of the fertilization. The sperm then faces the daunting task of penetrating the zona pellucida to get to the oocyte. Then evolution's response to this challenge is the acrosome, a huge modified lysosome that is packed with the zona digesting enzymes and located around the anterior part of the sperm's head, just where it is needed. The acrosome reaction provides the sperm with an enzymatic drill to get through the zona pellucida. The same zona pellucida protein that serves as a sperm receptor also stimulates a series of events that lead to many areas of fusion between the plasma membrane and outer acrosomal membrane. Membrane fusion, actually in exocytosis, and vesiculation expose the acrosomal contents, leading to leakage of acrosomal enzymes from the sperm's head. As the acrosome reaction progresses and the sperm passes through the zona pellucida, more and more of the plasma membrane and acrosomal contents are lost. By the time the sperm traverses the zona pellucida, the entire anterior surface of its head down to the inner acrosomal membrane is denuded. So this animation to the video over here that depicts the acrosome reaction with acrosomal enzymes. Sperm that lose their acrosomes before encountering the oocyte are unable to bind to the zona pellucida and thereby unable to fertilize. Assessment of acrosomal integrity of ejaculated sperm is commonly used in semen analysis. Penetration of the zona pellucida. The constant propulsive force from the sperm's flagellating tail in combination with acrosomal enzymes allow the sperm to create a tract through the zona pellucida. These two factors, motility and zona digesting enzymes, allow the sperm to traverse the zona pellucida. Then sperm oocyte binding occurs. Once a sperm penetrates the zona pellucida, it binds to and fuses with the plasma membrane of the oocyte. Binding occurs at the posterior, or you can say the post-acrosomal region of the sperm head. Fertilin that binds to a protein in the oocyte plasma membrane and may also induce fusion. Interestingly, humans and apes have inactivating mutations in the gene encoding one of the subunits of fertilin, suggesting that they use a different molecule to bind oocytes. Egg activation and the cortical reaction. Prior to fertilization, the egg is in a quiescent state, arrested in metaphase of the second meiotic division. Upon binding of a sperm, the egg rapidly undergoes a number of metabolic and physical changes that collectively called the egg activation. Prominent effects includes a rise in the intracellular concentration of calcium, completion of the second meiotic division, and the so-called cortical reaction. The cortical reaction refers to a massive of exocytosis of cortical granules seen shortly after sperm oocyte fusion. Cortical granules contain a mixture of enzymes including several proteases which diffuse into the zona pellucida following the exocytosis from the egg. These proteases alter the structure of the zona pellucida, including what is known as the zona reaction. Components of cortical granules may also interact with the oocyte plasma membrane. The zona reaction. The zona reaction refers to an alteration in the structure of zona pellucida catalyzed by the proteases from cortical granules. The critical importance of the zona reaction is that it represents the major block to polyspermy in most mammals. This effect is the result of two measurable changes that induced inside the zona pellucida. The zona pellucida hardens and the sperm receptors in the zona pellucida are destroyed. When the zona pellucida hardens, that crudely put, this is analogous to the setting of concrete. Runners of sperm that have not finished traversing the zona pellucida by the time in the hardening occurs are stopped in their tracks. When the sperm receptors in the zona pellucida are destroyed, therefore any sperm that have not yet bound to the zona pellucida will no longer be able to bind, let alone fertilize the egg. Following after the fusion of the fertilizing sperm with the oocyte, the sperm head is incorporated into the egg cytoplasm. The nuclear envelope of the sperm disappears and the chromatin rapidly loosens from its tightly pegged state in a process called decondensation. In vertebrates, other sperm components including mitochondria are degraded rather than incorporated into the embryo. Chromatin form both the sperm and egg are soon encapsulated in a nuclear membrane forming the pronuclei. Each pronuclei contains a haploid genome. They migrate together, their membranes break down and the two genomes condense into chromosomes, thereby reconstituting a diploid organism. Then the cleavage and the blastocyst formation starts. The product of fertilization is a one-cell embryo with a diploid complement of chromosomes. Over the next few days, the mammalian embryo undergoes a series of cell divisions, ultimately leading to formation of a hollow sphere of cells known as a blastocyst. At some point between fertilization and blastocyst formation, the embryo moves out of the oviduct or the uterine tube into the lumen of the uterus. The one-cell embryo undergoes a series of cleavage divisions, progressing through 2-cell, 4-cell, 8-cell and 16-cell stages. A 4-cell embryo is shown here. The cells In cleavage stage, embryos are known as blastomeres. Note that the blastomeres in this embryo and the 8-cell embryo below are distinctly round in shape. Early on, cleavage divisions occur quite synchronously. In other words, both blastomeres in a 2-cell undergo mitosis and cytokinesis almost simultaneously. For this reason, recovered embryos are most commonly observed at the 2, 4 or and seen here 8-cell stage embryos with an odd number of cells 3, 5, 7 are less commonly observed, simply because those states last for a relatively short time. Soon after development of the 8-cell or 16-cell embryo, depending on the species, the blastomeres begin to form tight junctions with one another, leading to deformation of their round shape and formation of mulberry-shaped mass of cells called morula. This change in shape of the embryo is called compaction. It is difficult to count the cells in a morula. The embryo shown here probably has between 20 and 30 cells. Formation of junctional complexes between blastomeres gives the embryo an outside and an inside. The outer cells of the embryo also begins to express a variety of membrane transport molecules, including the sodium forms. One result of these changes is an accumulation of fluid inside the embryo, which signals formation of the blastocyst. An early blastocyst containing a small amount of blastocellic fluid that is shown here. As the blastocyst continues to accumulate blastocellic fluid, it expands to form, you guess it, an expanded blastocyst. The blastocyst stage is also a landmark in that this is the first time that two distinctive tissues are present. A blastocyst is composed of hollow sphere of trophoblast cells, inside of which is a small cluster of cells called the inner cell mass. Trophoblast goes on to contribute to fetal membrane systems, while the inner cell mass is destined largely to become the embryo and the fetus. In the expanded blastocyst that's shown here, the inner cell mass is the dense looking area at the bottom of the embryo. Eventually, the stretched zona pellucida develops a crack and the blastocyst escapes by a process called hatching. This leaves an empty zona pellucida and a zona free or hatched blastocyst lying in the lumen of uterus. Depending on the species, the blastocyst then undergoes implantation or elongates rapidly to fill the uterine lumen. As mentioned over here, the developmental progression depicted above for a bovine embryos is essentially identical to what all mammalian embryos go through, including the humans. For example, between 5 to 6 days after the fertilization. The same stages would be seen in mice at about 3.5 days after fertilization. In addition to the morphological changes in embryo that I have described here, pre-implantation development is associated with that be called as an awakening of the embryonic genome. There is, for instance, little transcription in the embryos of most species prior to the 8-cell stage, but as embryo develops into morule, then blastocyst, a large number of genes become transcriptionally active and the total level of transcription increases dramatically. So after this fertilization, we will see next how the embryo develops in week wise, like first week development, second week development, third week development, all these things. So all these things have happened within the first week of the fertilization. Then from next class we will study about the second and third week of the development. So thanks for watching.